Welcome, everyone. Thank you for joining Continuous Learning with Bioguard. It is my pleasure to introduce our webinar speaker, Dr. Sushant Sadotra. Dr. Sushant obtained his PhD from National Tsinghua University and Academia Sinica, Taiwan. He has expertise in biomarker discovery and protein structural biology, with a strong background in investigating potential biomarkers for target diseases. Currently, Dr. Sushant works as a diagnostic product specialist with Bioguard Corporation. In today's webinar, he'll introduce the blood groups in animals. Recent advancements in veterinary medicine have expanded the use of animal blood products. Understanding blood types is essential for cell transfusion across species, helping to prevent complications like neonatal isoerythrolysis in cats. This webinar will cover the importance of blood typing and available methods for blood typing and cross-matching in clinical and lab settings. Without further ado, let's welcome Dr. Sushant.
Hello, thank you, Dr. Robert, for your kind introduction, and welcome everyone for today's talk. Let's now wait and let me share my screen and we can start. Okay. So I hope you all can see my screen. So as you know, today's topic would be the blood groups in animals. I'll try to give you more idea about how the blood groups are identified in different animals, as well as we will go into more details focusing on feline. So transfusion, the concept of transfusion in veterinary science started first in 1665, when Richard Lower, a scientist or a medicine practitioner, first did the successful blood transfusion in dog, where he, you can say, dogs using time. Sorry. By the course of time, from 1665 to early 90s, there were more studies were conducted to understand the transfusion of blood within animals. And during these research studies, they found that there are a lot of complications that are happening while doing the blood transfusion, as well as there are some immune components that get involved during these kind of situations. So these immune components later was identified as antigens that are present on the surface of our RBC's membrane. And through this further concept, the idea of blood groups emerges. So, for example, we, as you know, in humans, we have 4 kinds of blood types, and we say that human follows the ABO type of blood group. So yeah, in the same way, in animals also, the similar pattern is used to learn or to identify about the blood groups in different animal species. So in today's lecture, we will first of all learn about different types of blood groups that are identified in domestic animals, and to highlight out of those blood groups, which one are more important, as well as to dig into details of blood typing in animals. We'll choose today and study more details in canine, in cats, where we will discuss the blood groups in cats, as well as how transfusion reactions are occurring, and then certain specific conditions such as neonatal isoerythrolysis, how it affects the newborn babies. And lastly, we will use to identify the types. So going further, let's start with learning about different blood groups in different animals. So we start with the dog. So in dogs, they follow dog erythrocyte antigen style of blood typing, which is DEA blood typing. So in dogs, there are 7 prevalent blood groups that are found, named as DEA 1.1, 1.2, 1.3, DEA 3, 4, 5, and 7. So out of that, DEA 1.1 and 1.2 are identified into 60% of the population in dogs worldwide. Yeah. And DEA 1.3 is, for example, most prevalent or seen in German Shepherds, especially in Australia. However, the other blood groups are more rare to be identified in dogs, particularly, for example, in DEA4. It is most prevalent in the species that have already been transfused with the DEA positive blood. So in those dogs, because of the transfusion, those dogs will have this kind of blood group. The similar way is followed by DEA 3, 5, and 7. However, the reaction of sensitize of these antigens are a bit lower rate. Yeah. Next, in cats, they follow the AB blood group types, where 3 blood groups are identified in cats. So A is the most prevalent one, which is found, for example, in 95% of the American cats. However, we will learn more about the specific geographic distribution later on. Type B blood group in cats are basically seen high in Indian or Persian cats, as well as cats in UK. However, the AB blood group is very, very rare to be identified in cats. However, some of the domestic short-hair cats, as well as long-hair cats, are still being seen with AB, but the percentage is very, very low or negligible. Yeah. There is another recent, being done on the cats, and it was also identified that there's. However, this is a totally different topic to be studied, so we will not discuss this in today's lecture. Okay. So let's move on. So we go in the horses. So in the horses, you can see there are 7 blood groups that are identified, named as A, C, D, K, P, Q, and U. And with more than 30 erythrocytes antigen. That makes the blood typing in horses very, very complex, especially when you want to do inter blood group transfusions. However, blood group A and blood group Q are found in the most high prevalence, especially in the Arabian breeds as well as thoroughbred horses. And the QA antigen is still lacking in the standard breeds as well. Yeah. So talking about the cattle, there are 11 blood groups that are identified, which are A, B, C, F, J, L, M, R, S, T, and Z. And out of these, B and J are the most clinically relevant because those are the ones which are found in most of the cattles. However, B blood group is very complex because it has 60 antigens so far have been identified, so which makes the blood transfusion not an easy job. However, the J antigen out of these is a very special one because this is identified as a blood group. However, this is not a protein. This is a lipid which is found in the plasma. Yeah, so the characteristics of this component is different from other blood group types. So other livestock animals such as sheep and goats. So like in sheep, there are 7 blood groups that are there, which is A, B, C, D, M, R, and X. And out of these, the B blood group is the most complex one with 52 antigens that are identified there. And the R group, the AG are soluble and passively absorbed to erythrocytes, which makes it very rare as well. And the M group, for example, is again the special case where instead of acting as a blood group, this is also helping as an active transportation of the potassium ions between the reticulocytes. So similar to the the goat is also identified so far. Yeah. Okay, so this is all about we talk and discuss different types of blood groups in different pet animals that usually we also see in our reference lab at Bioguard. However, to go more details into topic, we pick Feline today to discuss this into more details. So as we know that feline are following AB blood group type system. So in this one, you may know that A, which means that the RBCs, the cell type, the RBCs have antigens that are there on the surface of the cell is for A. So this is named as A antigen. That's why the blood group will be A. However, in the plasma, it will let the formation of antibodies, which are anti-B. Yeah. So just to remember, it's easy, it's not that difficult, and very similar to the human blood group system as well. So like, for example, to know about the blood group B, which means that the red blood cell types will be of B in a way that they have an antigen of antigen B on their surface. However, in the plasma, they will have antibody, which is anti-A. Yeah. So lastly, there is a blood group AB, which as we notice that this is the most rarest one. However, if we talk about the characteristics of this will have antigen AB, both antigen A and antigen B on the surface. However, there will be no antibody in the plasma. Okay, yeah. So now, out of these three, how it is decided or how it is these blood groups are formed in the in the organism. So, for example, an enzyme which is known as CMAH, which is a cysteine monophosphate and acetyl neuramic acid hydrolase, is helping in the formation of these antigens. On the surface of the erythrocytes. So how does it do that? Hydrolyze glucose neuramic acid into acetyl neuramic acid. So acetyl neuramic acid is actually the antigen of type A, which when displayed on the surface of the RBCs, it is named as blood group A. Yeah. So that's why this enzyme is very much active in the most of the species, because as we know that most of the species in cats are blood group A. So which means the CMAH function is very much active in most of the cats. However, when the CMHA function is inhibited due to certain reasons, then the blood group B will show into scenario. However, in case of AB, the CMA function will be partially stopped or inhibited. So both antigens are produced by RBCs and displayed on the surface. Okay, so this is very easy to understand is because of the one enzyme that is controlling the formation of antigen on the surface of RBCs. However, when during the cell division process, how the organism is decided whether it's going to be A, whether it's going to be B, which means indirectly how the function of CMAH will be decided. It's based on the genotype or the cross matching of the two alleles that is happening. So, for example, here you can see in the table when there is an allele of A, parent A matched with the allele of parent B, second parent, I mean, but both the alleles are A and A, then the blood group gonna be, the phenotype of the blood group gonna be A. However, if one parent is A and one parent is AB, then of course the blood group gonna be A as well. And it is seen that when the parent is A and other parent is B, the blood group will be A. And the similar way, A, B, and B, yeah, this chart. So by conducting this kind of study in the cross-matching genotype and the expression of the genes, A allele is the dominant one compared to the AB and the B. Okay, and then it's also been seen that A allele is dominant to the B allele, and AB allele is to the A allele, but it's co-dominance to the B allele. So based on these, scenario, the phenotype of the blood groups come into existence in the offsprings in cats. Okay. So now, after that, we will understand the distribution of feline blood typing, globally. So distribution can be, we can subdivide into two types, which is non-pedigree, where we can calculate, where the studies been calculated on the overall cats present in a certain region. However, the second study also being conducted where a specific pedigree or a species being counted to see how many numbers of blood groups are there in those species. Yeah. So we'll start with some data to be shared with for non-pedigree species. For example, in the region in Asia, in Asian region, of course, A percentage is the most highest one, including Taiwan, where we are. We have 90% of the cats here with blood group A. Yeah. However, in Japan and in China, Israel, Turkey, for example, the A percentage is still the highest. However, some other countries, mostly in the Middle Eastern, close to the Middle East, such as Israel and Turkey, they still have higher percentage, some higher percentage of B blood group as well compared to the East Asian countries. So in the American subcontinent, as well as in oceania such as Australia and New Zealand, you can also see that the A percentage is still the highest one. That's including USA. So there are like two studies I have been kept here, which is still showing one is a 99%, and one is a 83% based on the number of, and as well as in Canada and Brazil. it's also the A percentage, the highest one. However, you can see in the Oceania countries as well. And so in this one, so you can see that, for example, in England, so the area of Scotland and the southeast, there's a vast difference between the two, where in the Scotland, A percentage is more higher than in southeast of the England, where A is only 67%, but the B is covering the rest of the 30%, as well as there are some percentage of AB blood groups also seen in those areas. However, when we go down towards the Portugal, there are also like 3 studies which were there in the on our database, where you can see the A percentage is still identified as the highest percentage present in this country. In other countries such as Spain, France, Italy, Northern Italy and Italy in overall, Croatia, Hungary, and Greece, you can see the A percentage is the highest one. Only I can see in Greece that the B blood group is also quite prevalent in 30% of the cats, as well as some of the region in Greece also show with AB cats as well, with 1.4%. Also in northern Italy, it's been reported that AB percentage is still there. So yeah, there are some studies, based on the years they were conducted, but of course if there are some recent studies out there, you can still follow to see the distribution of non-pedigree style of the fact sheets like these. Yeah. However, in the pedigree style, when we talk about basically specific cat species, so this is not very static, as we can see that different countries of the same species will have different kind of blood groups. It can be A, it can be B. So it's not something certain that we can, say that this pedigree follows only A or this pedigree follows only B. Yeah, it depends on the region. It depends on, I think, a lot of evolution. Through the UK in total. So there are like some of the cat species that I've been placed here, and you can see the percentage is not all of them are following a blood group. For example, in Burman, in Prussian cat, in Burmese, the B percentage is also being identified, especially in British short-haired cat. Yeah. So B percentage is 58% and A percentage is 39.7%, which is quite unusual when we compare to overall data. Yeah. And similarly, in Spain, you can see the same things is happening in the British short-haired cats, is seen with the 77% of A blood group, as well as B is only 22.2%. Yeah. Other than that, Persian and the Maine Coon cat is still A blood group is the most dominating one. So in Italy, of course, there is more number of pedigrees which are compared, and you can see that Cifix have shown that the B percentage is quite prevalent in this pedigree, as the best also have 33.3% of B blood group being compared, which is only 66.7%. However, in Ragdoll and Persian cat, you can see in Italy 24% AB blood group, which is unusual, is very rare. However, in these two pedigrees, the percentage of AB is high. So you can see that the So not any consistent, overall concept of the geographical distribution. However, overall, still the type A is the most common blood group that is being seen compared to type B, which is less in general, as well as prevalent. Prevalence of the region and the pedigree in cat. So there are a lot of factors that could be there for which could affect the blood transfusion actions in cats. However, the most understandable one are the functioning of the antibodies that are there in the plasma of the cat. So, for example, formation of alloantibodies in the plasma, which means that whenever there is a transfusion, for example, occurring, so when an individual is exposed with an external antibody, antigen, they will express an antibody in their body, right, to fight for that antigen, external antigen. So this is one of the most common reaction or factor that could affect the blood transfusion reaction in cats. Second is that allo antibodies that are already there circulating in the cat. So as we previously seen that whenever there is a antigen on the surface of the cat, for example, and the blood group type A would have A antigen on the surface, they would naturally have anti-B antibodies circulating in the plasma. So that's another factor that could affect the blood transfusion if it's not done correctly. So blood transfusion in cats in a easy language, if we can see that if the donor is blood type A, it can donate blood to blood type B, as well as it can donate to blood type A itself. Yeah. However, if the donor is blood type AB, it can only donate to blood type AB, but cannot to blood A or blood B because it has both antigens on the surface, right? So that's the reason. And blood type B, on the contrary, it can only donate also to blood type AB, but it cannot receive blood from A as well because it has anti-A. Blood. So that's the reason of blood transfusion reaction or cross-matching before you do any kind of blood transfusions in the cats. So, for example, here we can take an example where if the type A cat, which does not produce any anti-AB allo antibodies, circulating, which are naturally properly circulating in the blood, and which means that they can, receive blood from type A or type B, right? However, the type A blood contains high levels of anti-A antibodies, which may cause transfusion reactions in type AB because they have the type A antigens on the surface. So that's why the donor as the blood type B cannot donate to blood type AB, right? Even the blood type AB doesn't have anti-AB antibodies in its plasma. Yeah. So sometime if there is a incompatible blood transfusions reaction occur or accidentally happen, for example, the type A, if the donor is type A, is given to type B, so what will happen? So the type A blood will, when get mixed with the type B, after transfusion, the the complex of the antigen and antibody will start a hemolysis reaction in the cat, which is quite fatal. So the incompatible blood transfusions leads to the critical hemolysis in the cats. So blood transfusion identification of cross-matching is very important. Yeah. However, there is the condition in cat which is known as feline neonatal isoerythrolysis, FNI. So this is a condition usually is happen in the kittens, where the kitten, if for example the kitten blood group is AB or A, however the mother is B. So whenever the kitten start the feed on the mother in the initial they start acquiring antibodies through that form, or so they can absorb the molecules of IgG from the milk and enters into the kitten's blood. So indirectly, the kittens are getting the anti-A antibodies. However, naturally they are type A and type B, so which means that it will induce hemolysis in kittens, and if occurs in a higher rate, of course, it's quite fatal. So that's why neonatal isoerythrosis is very critical condition, especially in the facilities where the breeding of the two cats are being done. So that's the thing that in this paper, it's given a general idea of how we can avoid neonatal isoerythrosis based on the male and the female allele combinations that could be mixed and matched to make a perfect combination where the newborn baby, there will be no problem in having feed from the mother. Yeah. So in general, what do you say that naturally neonatal isoerythrosis is not that prevalent. However, as I mentioned that in the facilities where the breeding has been done, probably artificially, it can be most prevalent. However, this can be avoided if you can identify the alleles-based genotype and phenotype of the parents, and also doing the cross-matching for the offspring's possibilities, and then plans accordingly. Yeah. So let's now, so we had a concept of what are the blood groups in cats, as well as the prevalence of those blood groups in different regions throughout the world, as well as the factors that are affecting the transfusion of these two, of all the three blood groups in the cats. Now it's come to the point where we need how we can screen the blood donors in the cat. That's common methods that are used throughout the world. So, and most cost effective as well. So first is the immunochromatographic method, where the technique is based on chromatographic technique. There are immunocomponents that are embedded on the surface of the strip. And when the blood sample is put onto the strip, it will run over those immunocomponents, and it can some reaction will happen due to the sandwich of the two components, and the results are being identified through eye directly. Yeah. So these strips usually are incorporated with feline antiglobulin agents, which then get reacted with the immunoglobulins present in the blood, right? Yeah. So this is a very simple technique, also used in different kind of rapid tests as well. It's the antigen antibody sandwich method technique. Yeah. So this is very common as well. However, agglutination is also another common technique that are used, mostly used in and preferred by most of the labs throughout the world. In this, so the blood reaction also happened with a specific antibody present, on the surface of this the card that it's used to do that. So, for example, you can see here we have produced our own feline blood typing kit, which is also based on a agglutination method, where the card is being embedded with specific antibody, and it can react with the antigens of the blood to give you a particular reaction, where agglutination of the blood will show you whether it's type A, type B, or type C. Or sorry, no type C. Yeah. So last but not the least is the PCR based method. So PCR based methods are the most I introduced you about the alleles that are present in the genome of any organism, right? So similarly, in cats, there are locuses for A, B on the genomic surface. So once if you know about the basic PCR, you need to design primers to amplify those segments of the genes. So once you select the particular locus of A and B, in the genome, you can find the primers and amplify those segments of the where the AB locuses, A or B locuses are present on the genome. And after that, how you will identify that whether it's A locus or whether it's a B locus, you need to do a restriction and nuclease digestion of the PCR product. Now, where, for example, one kind of restriction enzyme will only can cut the A locus. If it's a B locus, it won't work. So the size of the DNA will be the same. However, in the opposite, if you put another restriction enzyme, which is focusing on cutting the DNA locus of for B, so at that point, it can only cut the genome into two parts. So the sizes of the two fragments would be different. And once you will run the gel on the agarose, you can see different kind of sizes for coming up when you use restriction enzyme 1, and different kind of sizes coming out when you use the restriction enzyme B. And based on the size calculation, you will identify very easily whether it's a locus or whether it's a type A blood sample you used or the type B, a blood type in the organism based on the DNA. DNA, of the genome. So in this one, yeah, so this is also very precise test. However, the above two tests, especially the agglutination test, is also very common and easy to identify in cats. Different species where you see the diversity, for example, effective to correctly identify the blood groups in those species. Yeah. So let's talk about the feline blood typing test in more details, as well as especially the agglutination test, where I would like to introduce you and give you the demonstration of our feline blood typing kit, which is one of the most liked product by the veterinarians in Taiwan and worldwide as well. So I thought that this would be a great opportunity to give you more idea about how easy and precise and fast the results can be generated. Yeah. So this test, as I mentioned, that based on the simple agglutination reaction where the heart is there, which is embedded with anti-A or lactins, and it can identify type A, type B, or it's AB. Yeah. And the most good part is that the kit is stable. The test stable is at room temperature. You can keep the test for 12 months. Is a expiration date from the date of manufacturing. So yeah. And the steps, the procedure are also very easy, which is that you need only a 500 microliter of the blood collected from the patient or the organism, which is cat. Then you put 3 drops on the card, and then put 1 drop of buffer that also comes with the kit, and then just gently mix it for 15 seconds, and within 2 minutes you will see the results. So let me give you a live demonstration of the kit and introduce the kit to you. Yeah. So please wait. Yeah, I'm right now in our reference lab in Taiwan. Yeah. So that's where I work. So here we do a lot of tests. We have our PC packaging here. This kit, the box here, you can see it's a very simple packaging, and inside you will have instructions how you can use the test, and all sample collection as well as all the procedure is well explained in the instructions. Yes. Other than that, the kit has the buffer that is to be used, and the droppers as well, which are used to, when you drop the samples on the book card. So next is the main thing, which is the card, which is having the, incorporated lectins or onto the cards, which are used. And also there are 2 sticks, the sterile sticks, which you can use for mixing the sample with the buffer when you put the drops. Okay. So yeah, so this is a very simple kit. So let me give you a live demonstration. So for today, I have prepared a fresh blood sample from one of the patient. So the sample collection is also very easy. You just need to collect the sample in an EDTA tube. So before doing the test, let me wear the gloves. Yeah. You might be wondering why I'm having ears of a cat. Yeah. So there are 2 reasons. One is that this is the Halloween week, and my talk fortunately happened in the same time. So, and also today I'm discussing more about our felines and the pets as cats. So I thought, let's do the theme in this way. Yeah. Okay. Yeah. So I hope you can see the kit first, where you can see we have a card. Yeah, so the card already have the labels as type A, type B, and controlled in there. Yeah, and also it says 1 drop of blood in each section, as well as 1 drop of buffer in each section. That's it. And you need to mix, and we will be using these sticks to mix. So since it's sterile, so I'll keep it like that. So let's take the blood sample first. So 1 drop of blood in here, 1 drop of blood in here, and 1 drop of blood in here. Yes. Later, we use the buffer, yes, which is also 1 drop in here, 1 drop in here, and 1 drop in here. After that, we'll use the stick. So now you can see we give you 2 sticks, and there are 3. So we will be using 2 sides for 1 and 2 sides for other. So very gently for just count 15 to mix the sample. Yeah. Like this. Yeah. And then you can use the other side to mix the other one. Okay. Yes. On the last one. Okay, yeah. So now you can see the procedure is finished. And now it's just that you need to wait probably 2 minutes minimum, but I can already see reaction. Later, I will show you 45% of the cats that are A, right? So that's why it says probably, the probability should be type A. Now to see, is it really type A? As I said, when the agglutination happens, as you can see here, for the type A, the agglutination reaction should happen in the well first, right? Here. And if it's type B, then the agglutination should happen in the middle well, yeah. And if it's a type B, then the both should have agglutination reaction. Sorry. Yeah. And if it's a false positive result, then you will see agglutination reaction, all the 3. Yeah, which means that there is some problem with the sample or the card. Yeah. So let's check the results now. So if you can come more closer, you can see the reaction very clearly, right? So agglutination reaction, you can see happened in first one, which is type A over here, and type B is clean and control is also clean. So this clearly showed you easily, an easy procedure, how fast you can identify the blood group. You can see very clearly that B, no embedded lectin or any other protein. Yeah. And in first well, you can see the agglutination reaction very clearly. Yeah. So I hope that you understand the importance of how important is to calculate or to identify, the blood groups in each animal, especially some of the animals which are, mostly we are having in our houses and also facing a lot of diseases. Sometimes they have to get operated. However, to successfully keep them healthy, blood typing is one of the key factors that vets have to use or have to identify before doing some of the which the vet can use easily, as well as rely on the results that are coming through our product. Yeah. So that is for today's talk. And also, I would like to inform you about our social media platform where you can get the further updates on all our all the updates that we do in our daily work, our new products, as well as our upcoming events, or especially the monthly webinars. Yeah. So please feel free to follow our Facebook, LinkedIn, as well as check out our website, which will be very helpful for your future. Yeah. So let's pick some questions from the Q&A section. We'll see, we have 8 minutes, so we'll try to discuss some of the Q&A. Let's do that. So Dr. Rita, in the recent years, rearing is being popular in Bangladesh. However, the testing of blood grouping of animals is not more common in Bangladesh. This presentation, okay, thank you so much. So for your kind comment. Okay. Could microchemiluminescence in certain cats population or hybrid influence compatibility of transfusion safety? Sorry, I don't have the name for this. Okay, for this one, I think the factors that we discussed here for checking the compatibility of blood transfusion in cats. I haven't read about microchemiluminescence yet. However, I check it, and also the people are asking for PPT. So yeah, we have your email addresses. So yeah, we will share the PPT with you, as well as if you have joined for more than 45 minutes, yeah, of course, you will receive certificate as well. So let me see if we have more questions over here. The type blood B has antibody anti-A. How do the anti-A develop in blood type B in the first place, considering the blood transfusion did not have occurred? Oh, okay. So this is the how the body functions. When the type A antibodies have anti-A, when the blood group is B in the organism, and the alloantigens naturally present blood, which are anti-A, is the function of the body. It already produces antibodies in A, but not in very large number. However, when you encounter the anti-A from outside, then those antibodies get into action more precisely, and then more anti-A will be there in the blood. Yeah. How I did like to ask about blood donation. Oh, sorry. This is now my. Why is EDTA the anticoagulant of choice for a blood sample for which a PCT measurement? Okay, so for the sample collection, for the agglutination reaction, as well as for the immunochromatographic methods. So these methods are based where the antigen and antibody, or the two proteins, are interacting. So when the agglutination already have happened, because of the anti-agglutination, the agglutination or the sandwich of the two proteins won't be forming. So that's why it's recommended. Okay, yes. So I think I have addressed most of the question, but most of the comments I can see are asking for the PPT. So I hope you have really like the PPT that I have prepared today. So yeah, we will be sharing with you. If you haven't registered and just joined, please, you can still write down your email address in the comment section or the Q&A section, and we will be able to send you the PPT after the webinar or in 1 or 2 days. Yeah. So I would still wait for 1 minute so that all the speakers, as I mentioned, if you haven't registered, we won't be having your email address. So please leave your email address. So we will send you the PPT. Okay, so there's one question that are there any findings on long-term impacts of transfusion of health on health outcomes? So I think for this part, because transfusion reaction when occurs, it's a very quick reaction of hemolysis. So these days, the vets are, already very advanced in, having to identify the blood groups. So I think when you go in a higher higher animals, large animals, so identification of the blood groups is more important because in the dog, in the cat, it become more and more easy, as I just shared with you, that there are so many products in the market that a vet can, in 2 minutes, will get the results in their clinic. However, for the large animals, it needs further testing to correctly identify the blood group. However, still, if the transfusion have occurred, even the the correctly identified blood groups and you did the transfusion, and after that the complication may start. Now, if their match is not correct, especially through the hemolysis, that the cells will start getting degraded and more antibodies will be generated, which start a lot of immune reactions in the body. So it's not a very slow process as well, which will be fatal for most of the organisms. Yeah, so that's why very much careful if they are. But yeah, I think we can close now. It's 9 p.m. in Taiwan. Yeah. So thank you so much for joining today and getting some information and knowledge about cat feline blood typing as well as for other animals. And in coming months, we will have more important and relevant topics for you guys. So please keep following our social media so you will get important updates timely basis. Okay, thank you so much, and I'll say bye-bye. Yeah.
Thank you for attending today's webinar, and thank you, Dr. Sushant, for the excellent presentation. We'll see you on our next webinar, and good night.